Welcome to this video on metagenomic analysis, where we will compare two core approaches: single gene surveys versus shotgun sequencing. Metagenomics is the study of genetic material recovered directly from environmental samples, bypassing the need for laboratory cultivation. This is crucial as most microbes are unculturable. It provides a holistic view of microbial communities and is vital for ecology and health. We classify analytical methods into two main strategies based on their genomic target. Single gene surveys target specific marker genes like 16 srRNA. Shotgun metagenomics, on the other hand, sequences all available DNA fragments in the sample. Single gene surveys target conserved marker genes present across nearly all members of a taxonomic group, such as 16 srRNA for bacteria. By amplifying and sequencing only this specific locus, researchers can establish a phylogenetic census of the sample. The marker gene workflow involves extracting total DNA, performing PCR amplification using universal primers, next-generation sequencing of the amplicon, and finally, bioinformatic clustering for identification. Marker genes offer key advantages. They are significantly cheaper than shotgun sequencing and benefit from established databases like Silva and Green genes. The process is also high throughput, allowing for simultaneous processing of many samples. However, single gene surveys have limitations. They can suffer from taxonomic bias due to primer design and provide low resolution, making differentiation at the species or strain level difficult. Crucially, they are blind to function, revealing only who is there, but not what they do. Shotgun metagenomics is an untargeted approach that sequences the total DNA extracted from a sample. This comprehensive sequencing captures the complete genetic repertoire, providing both taxonomic and functional insights. Shotgun sequencing breaks all DNA into random fragments, unlike PCR-based methods. This eliminates the need for prior knowledge of organism identity. It captures genes, intergenic regions, and plasmids, detecting viruses and eukaryotes alongside bacteria. Shotgun metagenomics produces massive, heterogeneous datasets. This requires deep sequencing to cover low-abundance species, leading to high computational demand for assembly, and often results in a large portion of unknown or hypothetical genes. The shotgun workflow begins with DNA extraction and fragmentation, followed by library preparation and high-depth sequencing. The final stage involves bioinformatic assembly or direct mapping. Bioinformatic analysis of shotgun data follows two parallel tracks. Taxonomic profiling identifies community composition, and functional profiling maps reads and contigs to metabolic pathways. Investigating function with shotgun data reveals the metabolic potential of the community, allowing for the identification of antibiotic resistance genes, biogeochemical pathways, and the discovery of novel enzymes. In terms of taxonomic resolution, marker genes are excellent for coarse classification. Shotgun sequencing offers strain-level resolution and can detect non-bacterial taxa, offering a much deeper view. While shotgun sequencing provides deeper functional and taxonomic insights, marker genes remain the standard, cost-effective method for rapid, large-scale biodiversity mapping. Choose marker gene sequencing when your budget is constrained, you have many samples for comparative statistics, and your main goal is simply to determine which organisms are present in the community. Opt for shotgun sequencing if functional potential is your objective, you require species or strain-level identification, or if you are specifically searching for rare genes or pathogens. Hybrid approaches combine the strengths of both methods. Long-read sequencing, like nanopore, is used for resolving complex structural variations. MAGs, or metagenome-assembled genomes, reconstruct individual bacterial genomes from shotgun data. The future of metagenomics points toward increased automation in bioinformatic pipelines and AI-driven interpretation of, quote, hypothetical genes. Ultimately, reduced sequencing costs are expected to make shotgun sequencing the default choice.